So one of again another really unique thing that we do is we are one of the only companies to offer a an autologous exosome product that can be used in the clinic. The autologous exosome product, it was derived from our knowledge of PRP. We know that PRP is often used in the clinics, and there's a lot of really great things in PRP, and one of the great things in PRP are exosomes. So we wanted to be able to take a PRP-like preparation, but instead of using the whole cocktail of cell therapy, we wanted to be able to isolate the exosome-only fraction and concentrate it into a multi-dose product that could be used in the clinic now. So that's what we did. We developed this process where we developed a PPX collection kit, which is really a blood collection kit. So in the kit, there's a butterfly needle, a 60 ml blood syringe, sodium citrate anticoagulant, and a blood collection tube. So on the day of PPX collection, the physician will use this collection kit to draw 60 mLs of whole blood using the syringe and anticoagulant. Then they'll transfer that 60 mLs of blood into our blood collection tube. That blood collection tube then gets put into a temperature controlled shipment box. So we use a 4 degree Celsius cold shipment box. That shipment box will then be shipped back to our lab overnight, where then we'll receive that blood the next day, check it into the lab, and then we process the blood to remove all of the cellular components. And from that 60 ml collection, we create 2 doses of around 600 billion exosomes per ml. So we're creating two 1 ml doses that are exosome precipitate in saline. So we essentially are removing all of the platelet component, all the red blood cells, all of the plasma component, and we're isolating that exosome fraction into saline and creating two 1 ml frozen doses that can be used later on. So after we create those 2 doses, we freeze them here in our -80 degree storage. But at that same time, we take a small sample of the product and we test it for microbiology and endotoxin concentration. So the microbiology test is a 7-day inoculation test where we inoculate the product directly into aerobic and anaerobic bacterial broth. That broth then gets monitored for a 7-day culture until we can ensure that the sample was sterile. So upon that end of that procedure, we're able to issue a certificate of analysis and ship the product back to the clinic for use. So from the point of blood collection to product return, it's about a 10-day period of time. So because this product is new and we're the first to manufacture it, we've put a lot of work into characterizing the exosome fraction to prove that we are in fact isolated and concentrated down exosomes. So in order to do that, we've done a few different nanoparticle tracking analysis experiments to count and really show that these nanoparticles are exosomes. Because exosomes are so small, they are typically around 50 to 100 nanometers in size. You need special instruments, and you need to use more than one instrument to really characterize what's in your product. So the first way we characterize it is with the NanoCyte machine. It's a nanoparticle analysis that's able to determine the size of the nanoparticles in your product. So the histogram on the left shows that our product has nanoparticles typically less than 100 nanometers in size, which is what we want. It's the expected size of exosomes. This histogram is also important because it shows that we've really cleaned our product, removed all cell and large cell debris. So our nanoparticle product is truly acellular and clean. And then the next thing you want to do is you want to combine in a fluorescent antibody that binds to a exosome marker. The exosome marker we use is CD9. So the right kind of shows you up front that if I stain these nanoparticles, I can visualize there are in fact exosomes within this nanoparticle population. So the next step, the final step is I combine those two techniques. I combine the nanoparticle size-in instrument with the fluorescent tag-in in order to specifically count the number of exosomes I have in my final product. So here I have three different curves. The black curve is just the total nanoparticles that are analyzed in this instrument. And then the blue curve is the total nanoparticles that bind to that CD9 antibody. And then the third and the last curve in the dotted line, CD9 is a antibody control. So that's just the negative control for the staining.
Mhm.
Right. So what the results show for this product is I had I had 4.8 times 10 to the 12 total nanoparticles. And from that population, 2.8 times 10 to the 12 were in fact exosomes. So I created a 58% positive exosome product. And I know that this particular dose had two 2,800 billion exosomes per ml, which is quite a lot of exosomes in our dosing scheme.
Mhm.
And for example, the average dose that we study in our clinical trials for reference is 200 billion exosomes per ml. That's the number that we've been studying in our amniotic fluid protocols. So with PPX, with this autologous, because I'm concentrating that 60 ml blood draw down to 2 ml total volume, I'm really
Mhm.
able to create a pretty concentrated product suspended in saline. All right, so then the next thing I do is we've been working on a lot of bioassay development in order to test the biological effect of this autologous product in the cell culture dish. So the one model we've developed is a macrophage model where we take monocytes, differentiate them into macrophages, and then we treat the macrophages with LPS1, I'm sorry, with LPS in order to stimulate an M1 phenotype. So we're able to stimulate the pro-inflammatory phenotype. And when you stimulate them, it upregulates the classic pro-inflammatory cytokines like TNF alpha, IL-1 beta, IL-6, and IL-8. So we look to see if we treat the macrophages with the autologous product, can we modulate and can we reduce the upregulation of these pro-inflammatory cytokines? So that's what these results here show. We looked at both IL-6 and IL-1 beta expression. So in red is the positive control. So when you treat with LPS, we get the 100, the maximal response of the pro-inflammatory cytokines. If you treat with PRP from a donor, so the PRP preparation is the mixture of platelets, plasma, and other cell components that we were isolating, we were able to significantly reduce that pro-inflammatory response. But as you can see with that standard error bar, it was a bit variable between some donors. Sometimes it worked really well, sometimes it didn't work so well, and sometimes there was no effect, but still significantly lower. But then if you treat with the isolated exosome fraction suspended in saline, so the cleaned up version, our autologous biologic, we had a very strong, significant suppression of the IL-6 and IL-1 beta. So this model has been really helpful for us to show that the various products we're making from donor to donor all have a very consistent anti-inflammatory effect with macrophages. And then the last piece of the puzzle here is we also do a lot of research to characterize the cargo of our different exosome products. Because again, exosomes are truly just a lipid bubble and the way they function is through the messengers they carry. So because all exosomes are made from different sources, they all contain different types of cargo and molecules that could make them more efficient for different indications or different usage than others. So we've been doing a lot of research looking at the microRNA cargo of various exosomes that we make in order to try to study which ones may be better for osteoarthritis or which ones may be better for hair, which ones may be better for the lung. So we do this through a concept we're creating called exosome signatures. So on this slide, I have three different exosomes that we've made. PPX is the autologous blood-derived exosome. We've made stem cell-derived exosomes from mesenchymal stem cells isolated from umbilical cords. And then we also have our amniotic fluid-derived exosomes. And here I'm listing the highest expressed microRNAs from the highest to, this is like the top 15 or so microRNAs that are expressed in each of the exosome types. So with the blood derived microRNA 16 is of course the highest expressed one, where that microRNA does not exist in the other two sources of exosomes. So we're using this to try to compare and understand what's unique about each product. And then from identifying what's unique, we can then try to connect it to different mechanisms. So this slide is an interesting publication that we found that tested the microRNA composition in cartilage samples isolated from patients with osteoarthritis versus healthy controls. And they identified that microRNA 26A and 26B were both significantly downregulated in osteoarthritis. And then they also connected these two microRNAs to cartilage dysfunction, and they claimed that these microRNAs assist chondrocytes in maintaining and developing the natural cartilage biology. So so part of the exosome therapy concept is if you can deliver back those two microRNAs that are lost, perhaps we could reestablish the normal biology of these cartilage cells and help repair the knee. So if I look back at those signatures that we analyzed, the plasma derived, the PPX, has both that high expression of 26A and 26B. The stem cell exosomes that I created did not have those microRNAs in its highest expressed cargo, and the amniotic fluid cargo only had microRNA 26B. So based on our hypothesis, connecting our data to the literature, we're able to form that strong hypothesis and idea that perhaps this blood-derived exosome product is really good for osteoarthritis because it has some of the key microRNAs that are needed for cartilage repair. I'm going to skip that one. So with that, we have done some small pilot studies. Aside from our large IND clinical trials, we also try to do a lot of small IRB clinical trials with our autologous biologic. So here's an example of one that we did where we treated patients with osteoarthritis with the PPX either intravenously or interarticular. And then we collected their WOMAC score data, which is the self-reported stiffness pain function score. And we looked to see how that score changed 3 months after the PPX treatment. And we found some good reduction in that WOMAC score when the patients were treated with PPX. So this was some of the just small data sets we have, but encouraging data to help us move further into really trying to use this product for musculoskeletal and other types of pain management treatments.
Did you guys see any difference between the IV and the IA administration in results?
No, not too much. If I separate out the graphs, it's hard to say because we had more IV patients make it through 3 months than the IA, and
Okay.
make it through in terms of follow-up. A lot of the IA ones didn't do follow-ups, so we weren't able to get good data to compare them. But it is interesting that we do see that IV has been working for a lot
Mhm.
of our osteoarthritis treatments. And our amniotic fluid exosome clinical trial is an IV route of administration, which also makes that protocol a little unique.
Okay.
Lastly, so in our product development, we've really tried to position PPX as the new alternative to PRP, and really have tried to use our research to highlight some of the reasons why we believe PPX is the new superior version of a blood-derived autologous biologic,
Mhm.
largely because we're able to concentrate a really therapeutic active component of the PRP and make it acellular. We produce the PRP in our FDA compliant and inspected laboratory versus through technicians in the lab. And we test our product for microbiology contamination, endotoxin, and we're able to do a lot of characterization to make sure that each product we make is reproducible, consistent, and effective.
In the
That's
last
it.
study that you had talked about with the OAI-IV, so you were talking about most of your studies have 200 billion exosome per cc dose. So is that
Yeah.
what was done there, or was it the
No,
600?
yeah. It would, so that one was more variable because it's autologous. We're
Hmm.
not controlling the exosome concentration as much as we can with the amniotic fluid trials. Because
Okay.
because amniotic fluid is a bit more reproducible in the way we make it to make sure that we always have 200 billion. But
Hmm.
with the autologous product, we're concentrating as much as we possibly can with each donor. So there is more of a variation of concentration donor to donor. So some donors have that really high 1,000 billion range, where some
Hmm.
donors have only a 400 billion dose range from the one we're able to produce. So dosing is a little bit more variable with the
Sure.
autologous, but still the way we've optimized our process to ensure that we can produce at least that 200 billion exosome per ml with the autologous product. But we're getting more, a bigger range from that 200 billion mark up to 1,000 billion.
And so I draw the blood, I send it. Do you guys send me a report with how many exosomes are supposed to be in that sample if we're doing autologous?
No, no, we're not doing that per patient. Per patient, we're only testing microbiology and endotoxin.
Okay.
However, if you would like to see those counts on a few of your patients, I can work with you to get that data, but
Hmm.
we can't do it like on every single
On every single.
donor. Yeah, just because it's a more costly analysis that
Sure.
takes time, but more than willing to, if you'd like to see what some of the readings look like, I can work with you to give you some data sets.
Or, what would be helpful for me is if I had some sort of gauge on, because right now with our PRP, I'll tell them this is the dose of PRP that you're getting, and this is how you're
Hmm.
going to do. If I'm just injecting 1 cc of exosomes and I have no idea how much there's in there, is it going
Yeah.
to work? Is it not going to work? Is there some sort of correlation with platelet number, platelet count that we're seeing?
Hmm.
Anything?
No, it's a great question. I don't have that data yet, but yeah, it's totally possible because if there's more platelets in the blood, it's very likely there could be more exosomes secreted,
Hmm.
from the collection process. But I haven't made that connection yet.
Okay.
But yeah.
So like right now, it's just, I mean, not to be a ball buster, but right now it's just I send the blood. We assume there's at least 200 billion exosomes,
Hmm.
maybe more, and then we hope.
Absolutely. Yeah. We know for sure that we're producing products with that dose range that we are actively studying. So we are getting exosomes 200 billion and above,
Okay.
and that is the effective dose that we're studying that has been shown effective in clinical trials.
Hmm.
But yeah, and yeah, so we also do ELISA methods to quantify things, which is a little easier.
Hmm.
So once you see experience where you have some, if you're seeing effects or would like to know that patients are consistently getting similar products, we can do ELISAs to show at
Okay.
least on that method, concentrations of exosomes are consistent, collections are being done properly, the product is as we expect. But yeah.
Are there any complications that have been seen so far with delivering too many exosomes, right? Because some
Mm-mm.
of these products,
Yeah.
they say 100 trillion exosomes, which is a lot of it's debris, right? Or
Yeah.
like
Hmm.
incomplete packets. If you actually put 2 trillion exosomes IV into somebody, are you seeing, am I going to hurt
Yeah.
somebody if I don't know how many I'm going to put in?
Yeah. I would say so far, no. I've never heard of that or seen that. I can all I can really speak to in that sense is what I've read in like preclinical trials with large animals.
Hmm.
So I know for sure exosomes have been treated in large animals up to like 5 times 10 to the 12. So that I would say between that 100 billion mark to 5,000 billion mark is what has been done in clinical trials or no,
Hmm.
in animal models. No, I have not seen clinical trials go higher than that.
Okay.
So the active dose range is really there. So I think anything between that 1 times 10 to the 11 to 12 is expected and normal and wouldn't have any negative effects.
Okay. What else was I going to ask? I had another question. I can't remember it.
No worries. So what do you use in PRP mostly for? Or you're mostly musculoskeletal,
I'm mostly
yeah.
musculoskeletal. I'm actually just down the road from you guys. I'm on
Okay.
State Road 84, in between Hiatus and Nob Hill.
Uh-huh. Okay, cool.
So, no, I do mostly musculoskeletal, but since it's just me in this practice, we do a little bit of aesthetics. So we do like PRP microneedling, PRP
Mhm.
facials, PRP injections in the scalp for hair growth,
Yeah,
microneedling scalp. But most of what I do is bone marrow adipose, and PRP for joint pathology.
great.
Yeah. A lot of knee and hip OA. I just saw somebody with an adductor tear, which is kind of fun. I'm gonna fix him
Uh,
so he can go back and play in the NBA.
yeah.
Um, I don't know. This question that I had is now fleeting.
No worries.
Oh, I guess so. It's frozen exosomes. So they come to me frozen. You send me two vials. How long do the vials last? Do I need to use them right
Oh
away?
yeah.
Store them?
Yeah, so right now our expiration date is 6 months for the autologous product.
Hmm.
And that's largely because we want to get the product out the door and have the doctors use it and all that. But we're going to be extending the expiration date for 1 year. Our amniotic fluid biologic has been validated for 4 years. And the
At
reason
what
I
temperature? Does it have to be at
uh,
minus 80?
minus 20. So it
Minus
just has
20
to be stored frozen in a minus 20 degrees freezer.
for 4 years, you said?
Well,
The
our expiration dates are truly based on our validation work and experiments that we've done. So
Mhm.
right now the PPX is 6 months,
Okay.
and I'm working with my experiments to prove that 1 month it's also stable, which I'm expecting to have no problem doing,
Hmm.
because our amniotic fluid exosome product, we've already done experiments to show that 4 years these products are stable when frozen.
Okay.
So exosomes in general are very stable product, and as long as they remain frozen, there should be no problem. But yeah, we typically use these PPX products within 6 months post manufacturing.
6 months. Okay. And the autologous is autologous, right? So I
Yep,
can't use it on anybody else, right?
correct. Yeah,
Okay.
correct.
Oh, that was the other question. So when you were showing me all the microRNA signatures that you're getting with the
Mm.
exosome, do we know what any of the other ones are doing in the platelet derived one versus the M? Or
No, I mean, not directly. Again, another example is like because I've been studying that LPS pathway, which is that toll-like receptor 4 pathway, there
Mhm.
are a good amount of them that directly target the that signal and pathway. I can bring that up real quick. You can Let me show you this slide. So yeah, so this is kind of a little slide I made up where I have these two publications that show the signal transduction pathway for toll-like 4 receptor. So
Mhm.
everything from, the receptor signaling down to all the signal transduction molecules to actual upregulation of the IL-6 and TNF alpha. So I put a little arrow here on those microRNAs that are known to target different pieces of this pathway. So even within this autologous product, we have high levels of LET7, that microRNA 26, and again, LET7 down here. And then we have microRNA 16 hitting these TAC complexes. So it again is that idea that there is a lot of microRNAs that could contribute to multiple ways of silencing the pro-inflammatory pathway. But I think one of my focuses is really this LET7 microRNA directly targeting the messenger RNA of IL-6, where I
Mhm.
think these microRNAs are really good at binding to the messenger RNA of these pro-inflammatory molecules and just causing degradation on the spot. So I
Okay.
think this LET7, which if I go back here, so that's microRNA and LET7 are really the highest, most abundant microRNAs in the product. So I think LET7 is a truly important component. of the exosome.
Okay. And even the platelet derived, it has a higher amount of let7. Oh no, wait, hold on, that's the wrong one.
Well, yeah, lead-7 is actually really high in a lot of them,
Yeah.
which I think again why exosomes in general, no matter what the source, they typically have a strong anti-inflammatory effect. So seeing lead-7 across is kind of a key to that.
Hmm.
So, but then looking at some of the unique stuff like microRNA 16 and then the high expression of both variants of 26
Hmm.
kind of adds that little extra uniqueness to the autologous product that might be why it performs well in that toll-like 4 receptor pathway.
And then always the pink elephant in the room. What about, so what are the rules using these products since typically exosome products are not
Mhm.
allowed by FDA?
Yeah, no, exactly. Yeah, we've been working in many fronts to really work with agencies to get accreditation and really kind of get that pathway more defined.
Hmm.
But we believe that the pathway for this is very similar to PRP because it's an autologous blood-derived product. The regulations are pretty clear that it's a very similar biological components product to PRP.
Hmm.
And the way we operate our lab, though, is more similar to a HCTP. The regulations of how we operate our clean room and our manufacturing facility
Hmm.
adhere to the regulations you need to make tissue and cell therapy products. So we actually are working with AABB to get accreditation in cell therapy type of manufacturing, but the regulation of the product type more fits with PRP, which is a more allowed to be commercialized and used because it's an autologous biologic. And even when we've met with the FDA, they are very focused on the autologous nature of the product. You know, and making sure the product is labeled accordingly to be used only by the patient it's derived from
Hmm.
is really the key to letting this product be allowed to use in a clinic. Compared to other exosome products like amniotic fluid or stem cells, they're from donors.
Right.
So that pushes the regulatory nature of it in a different donor qualification direction.
And how do
Which
you guys non-autologous products, how does the FDA approach that for you guys?
Yeah, well, we've been approaching it through our clinical trials. That's why we began our IND pipeline.
Mm.
So we're trying to commercialize the allogeneic through the clinical trial work, and then we're also doing more aesthetic work. So topical uses of the amniotic fluid is allowed and permitted.
Right.
So we've been spending more efforts developing our hair products using the amniotic fluid exosomes
Uh-huh.
and also kind of cosmetic type of products that combine the biologic where we're able to do topical use in that with that.
Gotcha. And minimal manipulation with the autologous product, is it just a centrifuge and
Yep. Yes. So there's no addition. The only addition is the sodium citrate to preserve the platelets, right? But yeah, all we're doing is we're centrifuging the product to clean and remove the cell component.
Mhm.
And then we also have a filtration step. The very last step is passing the product through a point 0.2 clean and filter that we're
Mhm.
really able to remove any cell debris and any cellular component. So it's clean, it's filtrate, centrifugation and filtration in order to concentrate that piece of the plasma that we're finding to be therapeutic.
Gotcha. Okay,
Yeah.
cool. What else? What haven't I asked? That's important that people
Um,
ask.
no, I think those are all really great questions. I think, yeah, from the science and the regulatory, those are the two big questions.
Okay. Um,
Yeah.
all right, cool. Peter, you were telling me about some educational thing in February.
Yeah, let Tammy speak to it. She is one of the well co-organizers, but indeed there's an event coming up February 7th, Tammy.
Right, February 7th, and we've got several throughout the year, but this one will focus on MSK.
Okay.
So, yeah, and sorry, I have a baby in my hand, so.
It's okay.
Yeah, and once you register for the conference series, you can attend all of them. So you're not, it's like $480, I think.
Okay.
And you can attend all of them, and up to three staff members as well.
Oh, okay. Cool.
Yeah. We held them last year, highly successful. Just really good information. So,
Yeah,
All right.
in those meetings, we do like some lab tours, and I really try to explain in detail all the different equipment we have and how we manufacture and test the product.
Mhm.
So even, yeah, being local, it's a good chance to kind of see how we do everything and how compliant our lab is, which we're really proud of.
Yeah. Are you guys on NSU's campus or just next to
Uh,
it?
we're within, yeah. So our lab is within their Center of Collaborative Research.
Okay.
So yeah, our clean room is on their research floor. So we kind of have our own designated space where we have our own research lab, like our dirty lab space where we do all product testing and our research stuff. But then our clean room is fitted within their research suites,
Mhm.
and we are able to manage and regulate that clean room independently from them. But that's where we do all of our product manufacturing inside there.
Gotcha.
mhm.
I guess the other question is cost. What do I talk to you, Amy?
Yeah,
Yeah.
I can
Yeah.
send you all the information on our products and cost. So I'll do that with the science book.
Okay.
And
Perfect.
along with our new hair products that we have.
Cool. I'm
Is
losing
it-
my hair, so don't want to end up looking like Peter.
Thank you, my friend. I love you too.
I love you so much.
Yeah.
Yeah, no, I'd be interested in all those products. Obviously, I'm most interested in the autologous stuff. I really would like to stay in the autologous space. I have
Mhm.
used exogenous exosomes and I still use them, but if I can have a very good source where I can trust where they're coming from and all that, I would be much happier than, the constant whack-a-mole that the FDA plays with these exosomes.
Right. And we'd love to have you as a provider doing MSK.
Yeah.
Yeah.
Yeah, what are next steps? So you're gonna send me some stuff and
Yep,
then
I'll send you everything. I'll send you even a new account form just to get your account set up and more information on the conference and yeah, and then just get back with me.
okay, sounds
Sounds
great.
good. This is great. Thank you. I will stop the recording.